You are listening to Professor Megan Workman. Welcome to this introductory lesson on core concepts for biology literacy. In this lesson, I will explain what is science, what is biology and biotechnology. We will examine experiments and variables, what separates a hypothesis from a theory, and I will explain the shared scientific language of a measurement called the metric system. Then we will distinguish features of living things that separate us from the abiotic non-living world, and we will end the lesson by examining levels of organic and biological complexity and discuss a concept called emergent properties. Science. Presumably, you are listening to this recording because you're enrolled in one of my biology classes and you are already considering pursuing science or one of its related professions like engineering or clinical healthcare for a career. And this is a great way to support yourself and your family. So I want to encourage everyone on this journey because science relies on multiple perspectives. Specifically, science will benefit from your unique perspective. Whether you decide to pursue biochemistry, neuroscience, planetary geology, animal behavior, nuclear engineering, conservation biology, physics, aerospace engineering, and so on, you have a home among scientists. Welcome. The first thing to understand about science is that it is based on a handful of assumptions. The first assumption of science is that an objective reality exists. This means that whether or not any individual understands it, reality does exist. This does not depend on individual beliefs. The second assumption of science is that among this universe of objective reality, humans can only understand some of it. So here I'm showing in the image in the bottom corner of your screen a person contemplating the universe. It's not difficult to imagine that within the whole universe there are things that our human brains are simply not capable of understanding. Science ignores those things. Science focuses instead on things that are knowable to human brains. Within this knowable part of reality, science is further limited to the sub-portion that is also testable or falsifiable. Another word for this is refutable. This means that if something is false, you can find out. So science is a tool for studying this part of testable reality only. It turns out that when we develop better and better technology, we can observe more and more of this knowable reality. Like when microscopes were first invented, we could suddenly observe very, very tiny microscopic organisms and cells. And so our observable part of reality was expanded. So as technology improves, this part of testable, because it's observable, reality that human brains are capable of understanding expands. And that expands the parts of the universe that science can be applied to study as a tool. Let me demonstrate this with an example. Consider the hypothesis, my mommy loves me. This is part of my knowable reality. I know this to be true. And there's no test for it. There's no amount of evidence that could be collected to prove it false. Therefore, despite it being true, it falls outside the realm of science. Contrast that with this second hypothesis about my mom. Mom feeds me. If this were false, I could keep a journal for a year, maybe more, and I could collect data on whether my mom ever feeds me. And if she didn't, I would find out. So my mom feeds me is in fact a testable hypothesis. I can collect data, and if it were false, I would eventually find out. Science refers to the process of finding these things out. It is the process of discovering objective facts about the world through testing hypotheses. So the scientific hypothesis that mom feeds me has a test. Did she feed me? Yes or no? If it's false, I'll collect some observations and they will reveal that that statement is false. But with the non-science hypothesis that my mommy loves me, there is no way to falsify this. I simply know it to be true, and therefore it doesn't fall within the realm of science. In other words, you cannot apply the scientific method to studying things that fall outside of testable, knowable reality. And science refers to this process of applying the scientific method to reveal objective facts about the natural world. All scientific knowledge was collected in this way, and so all information presented in our course was discovered through this same process. Science is based on observation and thought, and it works. The top images show discoveries that were made possible through the scientific process. The first picture shows a young child with the devastating effects of smallpox, which has a debilitated and often deadly disease that has been eradicated through vaccination efforts. The next image shows healthy crops. We're able to feed 7 billion people on Earth through the help of genetic engineering of agricultural products. And the next three pictures depict various technologies, including landing a man on the moon and returning him safely to the Earth. All of these feats were made possible by following the scientific method or applying this tool to purposefully test ideas. Biotechnology is a specific type of result of scientific inquiry. It is the purposeful exploitation of biological processes, usually genetic processes, for the benefit of humanity, either for profit or to help clean up oil spills or to make more cost-effective treatments for maladies, or to genetically modify agricultural products to increase yields or to improve the nutritional value of food that we eat. And if you'd like to learn more about biotechnology, I invite you to watch a Netflix documentary series called Unnatural Selection, which focuses on a recent discovery in the field of biotechnology called CRISPR-Cas9, which is a novel way to pinpoint sections of the genetic code that you want to alter it in a specific way on purpose to create the end product. This has been used to genetically engineer human children that have now been born. And so, as you might be imagining, there are huge bioethical ramifications, and we'll talk about this more throughout the course. But not all biotechnology is as scary as genetically modifying humans to some unknown, perhaps eugenic purpose. Here are some additional examples. In 1978, E. coli was first successfully used as little factories for creating human insulin that could be then used to treat diabetes, saving lives and improving the lives of type 1 diabetics. The bottom image shows agriculture, and as of 2018, 94% of U.S. soya, corn, and cotton was genetically modified. Specifically, it was herbicide tolerant and resistant to insect pests, thus increasing crop yield. also controversial. So science is a process for discovering objective facts about the world. It leads to all sorts of interesting discoveries and potential new benefits to society. And one of the initial steps of this process is to develop a hypothesis. So the scientist's job is to come up with a potential answer to a question. Through experimentation and many observations, you can test your hypothesis to see if you collect evidence to prove it false. If you do this over and over again, and other scientists are also trying to prove this hypothesis false simultaneously, and all of you collectively together cannot prove it false, eventually that hypothesis might graduate to a theory. In science, the word theory is reserved to describe a hypothesis that has been repeatedly tested and confirmed many, many times over. There's lots of supporting evidence, and therefore it is considered a fact. Example theories include the theory of gravity, the theory of evolution by natural selection, the germ theory of disease. Because scientists work in many different cultures around the world, they have developed a shared language in order to document their experiments and communicate effectively about precise measurements. And so next we're going to talk a little bit about this shared scientific language. When testing your hypothesis, you may come up with an experiment. Your hypothesis might be that something, A, influences something else, B. In the table, I've given some examples. So A could be height, and maybe height you predict influences calories burned per day. In scientific parlance, this cause and effect relationship that you are hoping to test has certain terms attached to it. So height is considered the independent variable. It's the thing that's influencing the dependent variable, calories burned per day. So if you wanted to test this relationship, there are a number of other factors you might want to hold constant to see if you can uncover a relationship between height and calories burned per day. All of the things that may influence this relationship that you might want to hold constant, we call controls. This might be how much exercise the person does. That would certainly influence the number of calories burned per day. You also might want to hold age constant. You wouldn't want to compare an 8-year-old child to a 35-year-old adult. You might also want to control muscle mass. So maybe people of the same height have different caloric expenditure because they differ in body composition. But if you were able to hold all of these relevant logical factors constant, then you could start to assess whether height influences calories burned per day. This is just one simple example. In any good experiment, though, there is exactly one independent variable that you're hoping to test. The thing that you're going to measure, the outcome of your experiment, is called the dependent variable, and everything else is controlled. These are the attributes of a good scientific experiment, which we will we'll practice many times over in our lab assignments for this class. In our experiments, we also need to agree to measure things in the same way. I can't be using feet measurement while somebody else uses meters, for example. So in science, internationally, one system of measurement is used called the metric system. The base units of the metric system are meters for height or distance, liters for volume, and grams or kilograms for mass or weight. Then prefixes are tacked on to these bases, so kilometer, hectometer, decameter, meter, decimeter, centimeter, millimeter, micrometer, nanometer. All of these prefixes adjust the size of the unit that you're measuring. So if I'm measuring something very, very small, I might use the unit nanometer. But if I'm measuring the distance between two cities, I would use kilometer. So the metric system uses base units and converts them to smaller or larger increments depending on the needs of the scientist using these prefixes, and we will practice this in class. Here under the written prefixes are their abbreviations. Notice under micro, that's a Greek mu, not the letter U. We've talked about science and some language shared among scientists for measuring and for experimentation. I also want to give you a quick definition of biology, which is when you apply science to the study of any living system, like a living organism or an entire ecosystem. In order to be a biologist, you have to first recognize what is living versus non-living. And you've probably done this in elementary school already, so let's make a formal classification checklist for living things. Here are 7 distinguishing features that all living things have in common. All living things can do this. So the fish in this cartoon example can do these, but the rock cannot. First, living things like the fish or you are made up of cells. All living things reproduce. All living things grow from an embryo to a new, more mature organism, or from an initial cell into a mature cell. They develop over time. Living things evolve over generations. Populations of organisms change over generational time. That's called evolution. All living things respond to stimuli. If I poke this fish, it moves. The stimulus is the poke. The response is movement. All living things harvest energy from the environment, then use that energy to perform cellular functions to organize their structures and to fight entropy. So energy is coming into the system to constantly rebuild cellular structures that would normally break down over time. So all living things use energy. And all living things regulate an internal environment that is separate from the outside world. The inside chemistry of a cell can be very different from the ocean in which that cell is floating. If we think about living things, we can arrange them into a hierarchy of organization. And this hierarchy of organization, if you think of the whole Earth, then you break it down into separate ecosystems. And within those ecosystems, there may be populations of different types of organisms, and each population is comprised of multiple organisms of the same species. And we keep getting smaller and smaller. Eventually, we get down to a point past the cell where there are these little structures, which we'll talk about in a future lesson, called organelles. And organelles are large complexes of molecules, and molecules are multiple atoms put together. Any level of organization below the cell is considered inert. It's not part of the living world. And so life is considered an emergent property, or a characteristic that is only observed at a certain level of complexity and higher. So here at the cell level, life is sparked. It is the emergent property at this level of organization. When you're reading your textbook for this class, you may read about additional emergent properties that are only observed at the tissue level, or the organ level, or the organism level. So what have you learned in this brief lesson? You learned about what is science. You learned about 7 distinguishing features of living things. And you learned about where life begins within these levels of organic and biological complexity.